Hi, my name is Tomas, and this is my gut microbiome presentation. A microbiome is a community of microorganisms such as bacteria, fungi, and viruses that occupy a specific environment. They may be found in or on the human body or other organisms. A gut fecal microbiome is in the intestines of the body, which is home to trillions of bacteria, fungi, and other microbes that are involved in some important processes such as the digestion of food, regulation of the immune system, as well as fighting off other bacteria. One of the conditions we looked at in our experiment was weight and the relationship it has with the gut microbiome. In a study done by the NIH, they made observations between the two. The researchers used genetic sequencing of fecal samples that they obtained from 12 obese and 5 lean volunteers in order to look at the different strains of bacteria. The results showed that the obese individuals had more firmicutes and nearly 90% less bacteroidetes than the lean individuals. In addition, the obese subjects changed their diet to low fat or low carb for a year, which led them to losing almost 25% of their body weight. Not only that, but the proportion of firmicutes dropped while bacteroidetes rose. Another study showed similar results involving the same bacterial phylum, where there was a greater percentage of firmicutes than bacteroidetes for the subjects who had higher calorie intake. So both of these studies didn't conclude that there was a direct causal relationship, but rather a correlation relationship, where further research must be done. Another study looked at the relationship between diet and the gut microbiome. This was another study done by the NIH. Researchers used 16S ribosome gene sequencing in order to characterize the human gut microbiota into different enterotypes noted by the bacteria that was present. The enterotypes are strongly linked to long-term diets, specifically those with protein and animal fat. The results showed that protein and animal fat were associated with bacteroides, whereas carbohydrates were associated with Prevtola. They also looked at the controlled feeding in 10 subjects and noted that there was a change in composition of microbiome after initiating high fat and low fiber, and vice versa. The results allowed them to conclude that diet is highly associated with enterotype partitioning. Multiple studies have shown that the high-fat diet can lead to gut microbiota dysbiosis. This wasn't a direct causal relationship, but it shows a correlational relationship between the two. In our experiment, we looked at the bacteria in the gut microbiome of 4 dogs, 2 of which were male and different breeds, and 2 of which were female of different breeds. 2 of the dogs were obese, and the other 2 were of normal weight. The question we asked was, will the size and weight of the dogs affect the gut microbiome? We hypothesized that the 2 dogs that were overweight and of different breeds have more similar gut microbiomes than the 2 dogs of the same breed. For our methods and material, we had samples that were an overweight pug, a normal weight pug, an overweight German Shepherd, and a normal weight German Shepherd. For the experiment, we had to first isolate the DNA, then quantify and normalize the DNA, and finally analyze the quantity of specific bacteria or fungi within the fecal DNA by conducting a quantitative polymerase chain reaction. First off, following the instructions in the lab manual, we used a Chi-Gen kit to isolate the DNA from the feces so that we could get the concentration and purity. To get the concentration and purity, we used a nanodrop spectrophotometer and recorded the concentration and purity for each sample. To get the purity, we recorded the 2060 to 2080 ratio, and as long as the ratio remained between 1.5 and 2.0, we knew the purity was adequate enough. To normalize the DNA, we just calculated the amount of eluted DNA we needed from each sample to dilute it to 1 nanogram per microliter, and mixed it with nuclease-free water to make 10 microliters of the sample. We then mixed our diluted samples with 2x master mix, controlled DNA, which was E. coli, for our positive control, nuclease-free water, a forward and reverse primer. For the first round of qPCR, our forward primer was bacteria, and the reverse primer was fungi. The second qPCR, our forward primer was acidobacteria, and the reverse primer was actinobacteria. And for our final qPCR, we used firmicutes for the forward primer and Microbiome for the reverse primer. Once the qPCR strips were prepped, we gave them to the professor to be placed in a thermocycler. The results for our UV spectrophotometry showed that we had adequate amount of DNA in our samples because all of our values were above 1.0 nanograms per microliter. And the 2060 to 2080 ratio showed that our DNA samples were pure enough and free of contaminants because the values stayed between 1.5 and 2.0. For our first qPCR, which was bacteria versus fungi, the positive control for the bacteria had a CQ value of 15.13, which makes sense because we use bacteria as our positive control, so it was abundant, whereas it was rarely abundant for our qPCR that had the fungal primer. The negative control was successful for the qPCR for both bacteria and fungi because all of our CQ values and antilogs fell below the negative control values. For our second qPCR, we used acidobacteria and actinobacteria. And although the negative control and sample CQ values were all rarely abundant for the acidobacteria, we can't really trust most of the values because they did not fall under the negative control CQ value. Whereas for actinobacteria, most of the values were under the negative control value. This could have been caused by a contamination in the process of transferring the sample DNA to our qPCR strips. For the final qPCR, we used firmicutes and Microbiome as our primers and for both of our primers, the results for the samples were successful because all of the CQ values fell under the negative control value, although all samples for the bacteria were rarely abundant. The data we obtained did support our hypothesis because in all of our qPCR results, it showed that the overweight dogs, although from different breeds, did have closely related microbiomes versus the 2 healthy dogs. For example, in our first qPCR with the bacteria, the 2 healthy dogs had lower CQ and antilog values than the 2 overweight dogs. If we were to conduct this experiment again, we would like to try next generation sequencing because it would be able to tell us exactly how and because it would be able to tell us exactly what bacteria or fungal microbial DNA is present and how they would differ from each sample.